So today, one point in common that we have with Claudia and several others here, like Jordi, is that we are really interested in working with island systems. And I will first introduce a bit why we are so interested in islands and a research project that I started almost 1 year ago already that is called Timelines, that is Island Timelines to Quantify Biodiversity Change. So basically, this project that was funded by the ERC, what it aims is using all these islands that we have across different oceans, from the Atlantic to the Pacific to the Mediterranean Ocean, etc., taking them as model systems to basically try to use them as sensors or thermometer, if we prefer, to see how, where, how fast, why biodiversity, mainly understood as floral biodiversity in this talk, is changing. So this is basically to see how different are islands between them. It's not just that there are Arctic islands, subantarctic islands, Mediterranean islands with a different history, but as well, there is very different geographical features in all of these islands. For instance, this is Teide Mountain. We all know that has this massive peak, allowing a very nice study on biodiversity change along an elevation gradient. But then if we go to Mauritius, it's quite flat, tropical island with very different geographical characteristics. And for instance, this one that I will be explaining a bit of the results, this is a small island in Cabo Verde. This island is called Brava. And the picture is really taken from an island next door, let's say, that is called Fogo, with a massive volcano that has produced several impacts, not only in Fogo, but as well in Brava, because it was pushing a lot of people from this island to the little island of Brava, with all the potential impacts happening in this small, flat, arid island. So the essence of this project is basically trying to identify and trying to see the severity of all these impacts that I call here bottlenecks. But basically, what I mean here is that when people arrive in an island, which is the real impact that we can see in the landscape, it is maybe tropical islands responding different than maybe Arctic islands to the arrival of people. Is the toolkit of people arriving in the island that has an impact, or the amount of people arriving? Or maybe it can be that they bring different species on the island system. They may come with different crop species, but as well rats, rabbits, etc., etc. So if we put all this in a mix and we scale all this information up, and we just try to get this global picture with these massive gradients, maybe we can start answering, just to start answering a little bit, how vegetation is actually changing. If, for instance, the first question here would be, are rates of vegetation compositional change consistently maybe accelerating after initial human arrival? That said, in other words, would be when people arrive in an island, can we actually see a turnover of change? Is changing the vegetation around fast or no? Or is this change happening across the different islands that we have data? And this as well, should we start thinking on, is, for instance, this change happening different with native species than non-native species? Or another question that we can start answering if we integrate all these data sets is the second one. Okay, so what trends are plant communities following? Are they getting similar over time or no? So all these people that arrive in different islands, maybe introducing the same species, can we actually see any similarity trend in between them? So I will start with trying to show examples of how we are approaching, it's definitely not finished, but how we are approaching these two questions using paleoecological data from different islands. So you may think, okay, there's a lot of dots here, but maybe we are mixing up different oceanic islands, continental islands, different types of different islands. But this is already the data set available for all of you coming from the Automa database. These are paleoecological records only using pollen data sets that they're available out there in the Neotoma database. So for us, that was a good start in a way that to be able to approach these two big questions. And this is just an example of how beautiful are these pollen grains, and they in particular from the Canary Islands. So as all of you probably know, thanks to these morphological differences of all these pollen grains, we can identify which species, if we are very lucky, species, if not so much genus or family, but we can have a nice taxonomical resolution of what happened on a site and how much it changed the vegetation over time. So as well, we can have a little bit of the vision of how the plants look like. So we can start as well introducing a little bit of information on functional types and functional traits. So we pull all this together, and we now make an effort to narrow down to one case study. I will start showing you how the first question was inspired from. So this is the same picture I showed before, Cabo Verde, taken from Fogo, pointing to Bravo. This is a small dry island, and we know from historical records that it has suffered some migrations from Fogo to this island, with all the impacts that they were mentioning, that they were lost several land cover types, different land cover types, and basically what we know as well, apart from volcanic eruptions and the impact of migration, we know that here it went probably 5,500 years ago. There was a big shift in climate, like towards drier conditions. That would be the end of the African humid period, and these islands are quite close, so it's not well finalized, the impact of these regional climatic change, but something to take into account. Small, dry, quite heavily populated islands. So when we pull all the paleoecological information that we have from this island, and this is work from Álvaro Castilla Beltrán, and this is literally the field site in Brava, but we saw something quite interesting, or at least we thought that was quite interesting. This is an ordination analysis, and each of these points, that is different colors and different shapes in here, means how a community of vegetation look like in the ordination space. So if we follow from the past to the present, and we start here, this is a record that started more than 9,500 years ago. We see that these two samples on time look quite similar, but then literally something happened here, and the community nowadays looks like this. And if we look at the record, is when people start settling on the islands. So that was a suggestion telling us maybe there is a real impact on when people settle an island, but it's because it's a small island, or because there is a characteristics on this island, or is something quite big that we need to test globally. So we start looking at the literature. And there's a beautiful record from this island, from the Poor Knights archipelago offshore of New Zealand. And the focus of this study was this plant here, the Metrosideros, with these beautiful showy flowers that was targeted for restoration and for basically using it for restoring vegetation sites in different islands around. Okay, but thanks to the paleoecological records, they found that this was introduced, this plant, 100 years ago. So that was quite interesting as well to read, but this is not what I wanted to explain from this record. But basically, when we looked at the ordination analysis from these records, so the same analysis that we did in Brava, we start seeing a kind of similar trend in a way. So the people arriving in the islands, a community of plants, is basically in this ordination space. It was moving when the Maori gardening zone appeared on the islands, and then the European zone. So again, it looks like each different ways of colonization on the islands is leaving a footprint in a way on the vegetation communities on this island. So we started thinking if eventually all rates of vegetational compositional change consistently accelerating after initial human arrival is something that is globally happening across the different islands. And after a lot of formalization of data sets, trying to get the best data sets out there that allow us to compare what happens before and after people arrive, we selected these study sites, as you can see here. A little bit of space around. You saw, if you remember the image of the map full of blue dots, zoom. We ended having well, a few amount, but not as many as before, because we needed to really be quite thorough in the selection of data sets. It needed to be enough data points, etc. So you see that we have an ice spread in the Atlantic, Mauritius in the Indian Ocean, and as well a water spread in the Pacific. So to try to do that, we need to harmonize the data sets. And we followed a kind of quite straightforward, I would say, measure of change. So we use only the fossil pollen data, in this case, not the cDNA. There's not enough data sets for now that we could do that. And we use the percentage data. Then we were able to basically, you see the reconstruction vegetation history, and then is when the statistics come. And each of the scientists, they may have different approaches, but in our case, we focus on the measure of turnover. However, rate of change or different measurements could be as fine. To basically see what happened when people arrive. Is the vegetation changing or really is not? And with a team of biogeographers, archaeologists, paleoecologists, macroecologists, we start discussing these 4 years before the publication, or even further down in time. And we managed to get this publication published. And what we found was this. So for each of the data points I showed before, we did this measurement of vegetation composition on turnover. Basically is getting the ordination analysis in the shape of a slope. And what we can see here is that in most of the records you see here, for instance, in Española, we see that definitely when there is, if you focus on the orange vertical line, you see that in most of the islands, there is a kind of acceleration, an increase in the turnover towards the present. But there are very good examples, like they don't happen, that they are not following the general trend. For instance, now clearly is not following the trend. If we go to VT level, if somebody helps me finding it, neither happened. This is in Fiji. So what is showing this, apart from being a very busy slide, what is showing is that almost all the islands, when we analyze, when archaeologists, historians are showing us that people were settling the island, the turnover is increasing. You may wonder what it means, this dotted line. What we did is running the same analysis, but using when the data set is telling us that there is a change. Okay, so basically we see that what it was very good for us as well is that it's not statistically significant whether one of the lines happened before or after. It's pretty random. Okay, and most of them are separated maybe 100 years, etc. Although there are big examples like this one. So it's showing that is not because maybe the data sets were not dated well, or there is any methodological issue. So maybe the question was already answered in here, like say, yes, there is a change and acceleration happening across the islands, but we pulled together all this change and wanted to quantify eventually we have a big amount of change after people. And we pulled together all these data sets, and what we found is that after human arrival, rate of turnover accelerated by a median factor of 11. That doesn't mean that there were no changes before people. There were. We can think about different natural hazards, different changes, variability. But when people settle the island, this goes up to 11 times more. Talking about turnover, talking about change. So when we submitted that, the paper ended in here. Reviewers were probably not super convinced about that, and they asked us to think a bit more. So they asked us literally to put a little bit of context, geographical context, biogeography in our study. Basically, they wanted us to answer influence of geographical covariates. Is literally this change because of people were settling the island, so time since colonization, or it can be explained about the latitude, it can be explained about the isolation, maybe the elevation, the site. So the elevation of the site, you never know. So we tested that, and the results were these. As you can see, there's not a lot of significant trends in here, but the first that is human arrival. So that was, we think, enough to show that really the change after people arrive is overtaking, let's say, the variation, the change before people settle the island. But there were as well some other conclusions that we need to explore even further. That is in the first box on the top, that we say that differences between the pre-human and human-dominated turnover rates are systematically linked to human arrival times. And I think that this is quite interesting, and we haven't explored it enough. With faster rates on islands colonized in the past 1,500 years than those colonized earlier. So it looks like the change is much faster right now than even if the island is colonized 3,000 years ago, that, for example, the Polynesian islands. Okay, and clearly it looks like environmental conditions are not directly related with the data set that we have currently to this acceleration of turnover. So this would be the first example of all these potentiality of getting together different island data sets to answer, in our case, these questions that we are interested in. And this would be a little bit the conclusion. And as I already mentioned, the first 3 ones, I think that we could eventually discuss later. Point 4, that says that our results show little indication that these human-affected ecosystems are either similar to or returning to dynamic baselines. One thing I would like to highlight is that we're not talking about extinctions here. We don't know, okay? Or maybe the change is not as big as it looks in the turnover, okay? And this was something that was worrying us. If we could explain if this change was triggered by an introduced species, or this change, or the native species in declining. So we didn't know at this point. And I always like working with illustrators. And this is a work done by Jagova, a great scientist and illustrator. And we gave to him the paper, and he summarized it in this way. So these are 2 islands basically talking about people arriving to the island. And it says, yeah, they've been around for a while. And asking to the other, and yours, nah, barely a couple of 100 years. And I think that summarizes quite nicely this aspect of the importance of knowing for how long a place has been basically impacted by people. Work from Jagova. So linking with this, what we were thinking is that, okay, we know that is changing, but we clearly don't know which is the main trigger of this change, if it is the native, non-native, etc. And we know that this is quite an important topic because islands are hotspots of non-natives. So there are even studies that I will show a little bit later quantifying the amount of non-native species in each of the islands. So what we wanted to try is going a little bit further on time and complement these beautiful papers by biogeographers in this case. The first one, this one here, that says drivers of the relative richness of naturalized and invasive plant species on Earth. They already said that a total of 26 of all islands have already accumulated more non-native and native vascular plants, clearly putting quite a big weight on the management of island biodiversity and non-native. But the other one, and I think that this probably is interesting for all of us interested in the past, is that they said that the redistribution of alien species across the globe accelerated with the start of European colonization. So I was quite pleased to see that they are starting putting history on their analysis. And they said that the legacies of European colonialism can still be detected in today's alien flora, making that more similar. To say that the longer a region was occupied by an empire, the more similar the alien flora are still today. So that was really, I found it really interesting. And in that point as well, I linked all this information with one of the probably first papers that put a little bit the fallen records serving conservation in a way. And this is a study published already in 2008 from Galapagos. And this beautiful flowering here, that species, the hibiscus, it was listed basically to be eradicated from the islands because they thought that was an invasive species. They had their own reasons to think that because they were expanding the ranges, so very different aspects. However, thanks to this paleo record, 8,000 years ago, viscous was there. So maybe some of you would say, well, was this only pollen, maybe blown from another island, or maybe, well, they did as well macrofossils that they found the seeds. So I think that that is a very beautiful paper where paleoecology can be merged as well with conservation. So, of course, after this, we have many questions. Can we actually use our data sets to explain a bit more the diversity and dynamics of non-native species? For instance, how many non-native species are really native, inspired by, I think, always thinking of the modern checklist. So, you know, or for instance, appropriate species classification, or native or really introduced, or when did they arrive, what is the rate of spread, or the last one, for how long islands have been accumulating on native taxa? Is something only from the last 100 years that we know, archaeologists, we know that this has a longer history, mainly as well in different islands, or longer. And this was a work done and led by Anna Valentovitz, now postdoc at Bayreuth University in Germany, and Manuela Steinbauer, long collaborator as well, biogeographer with huge interest on history and paleo in general. And we basically got all that data sets I showed before from all those islands. And Anna did an amazing work of checking all the modern checklists, all the pollen records, and she harmonized the data sets. When was possible that that species was non-native? Non-native. If the taxonomical resolution of the pollen grade didn't allow us, we put it into another mixed category. And after following all this process, we got this. So basically, imagine it's the same data sets, but now separated with non-native and native. And this is basically the two harmonization lines. Okay, so this would be light red of the non-native only. So the species that we were sure that the equivalent of the species of pollen was as well non-native in the checklist, and the other one when we were not able to do that. So this would be the two ways of harmonizing the data sets. And as we can see, more or less around here, the non-native were already accumulating on these islands. It opens up to a lot of questions, because it can well be that a checklist, then there is a kind of, it's difficult basically to put a species into different categories, or that we really need to study a bit more as well the dynamics of non-natives on islands. This is the a little bit what we found on non-native. But then, of course, when you finish one thing, you start thinking on the other one. And then we thought, okay, if all these non-native accumulating for 1,800 years at least, is this as well showing any similarity trend across gradients on islands? So is this non-native species introduced in different islands producing similar changes or not? Or basically is increasing richness instead? So, well, we start thinking a bit on that. And this is the context of the last example that I have prepared for this talk today. And we wanted to answer a little bit. We didn't answer. We started trying to answer a little bit this question. Are islands getting similar? Or these non-native species basically trigger a trend in similarity? So why were you worried about that? Because apart from getting more similar, that means that probably is getting as well this kind of process of floral homogenization with all the consequences around. And basically, one of the first times that this concept of homogenization appeared is quite a long time ago. According to what we've been reading, is from the 99 already. Probably there is even older reference in here. And they say that the erosion of the biotic distinctiveness of communities driven by endemic species loss and introduction of non-native species can result in what is commonly referred to as biotic homogenization. And one of the latest one that I think that shows as well the importance of this concept is the text in the gray book, should be a gray there. They say that biotic homogenization is now considered one of the most prominent forms of biotic impoverishment worldwide, and it will likely continue to increase in response to anthropogenic forces. So we thought, okay, let's start first with a system that we really know if we see any trend towards similarity. So with the team in the Canary Islands, University of La Laguna, that we've been working in the Canary Islands for a while, and as well Cabo Verde, we pulled together the data sets and we basically start thinking, if there is any change, let's see if the communities start getting similar. And what we saw is this. While the two records on the Canary Islands are changing, following the same that I was explaining at the very beginning, all the sites that we had at that moment in Cabo Verde are kind of towards the present, getting kind of merged in the same quadrant, in the same space of the ordination space. So we thought that this was as well indicating us, and maybe we are starting seeing that basically this process could be tested or quantified using islands. So then we went a little bit bigger in the gradient. And with Nicola Strandberg now working at Tromsø, at Inger's lab, and as well the team of researchers that we normally work together in terms of biogeographical scales, we thought, okay, at that point, Nicola was working in Vanuatu on a core, and the pandemic started, and then the labs were closed. And discussing a little bit, we thought, okay, would you really like to learn a little bit of data analysis and try to get these analyses moving forward? And she definitely said yes, and started analyzing all these data sets. The data sets inside the box, her own data sets, and as well the Neotoma, the database data sets. And we know that this is something that it's relevant. It has been discussed and introduced this concept very recently as well. And the Pacific Islands in particular were highlighted in this global paper, saying the Pacific Islands show the fastest increase in species number, suggesting that potentially they are sharing a lot of different species in different islands. So there we go. Maybe some of you would think, why the Pacific Islands? So according to a lot of different researchers and literature, they say that the colonization of the Pacific Islands represent the last great migration of humans into occupied lands. This is what I read. If apologies if you find another example, but it's one of these, because starting more than 3,000 years ago, as you can see here, and then expanded all across the Pacific to Easter Island. Some papers suggest that it's 700. So there's still a lot of research going on these islands to really nail down and narrow down the time. So you can see all the different, in a very simplified form, the different migration of across the Pacific. So again, all these islands are quite different. From west to east, there is a gradient of 8,300 kilometers. Then we have islands in the tropics, subtropics, and warm temperate South Pacific type of climate. Elevations, there are islands that we have, of course, on sea level. So basically coastal areas, but as well, we have islands at sites at 760 meters above sea level. So allowing us as well a nice comparison, although limited comparison of what happened on the highlands of these islands. And then if we look at the different regions, we had 7 data points in Melanesia and 8 sites in Polynesia. And we cut our data set at 5,000 years for many reasons, but as well for statistic limitations of the time beings and the amount of data sets that we have across the different cores. The first result that we found is this, and we decided to plot it in bar charts. And what we did is basically comparing all the different time beams across all the time beams of all the islands included in the analysis. Okay, and after doing that, we color code when the analysis was showing that the trend was that time compared with the other was similar, so homogenization, or was keeping the difference. And as you can see here, Jakarta is all blue. So all the different time beams compared with all the islands are showing that they are getting similar. But there is huge exceptions, like this island here, or like this island here. So what is showing is this trend. But Nicola, as well, as started pulling together all the data sets again with a shape of a trend. That is how we really understand a little bit better what is going on. And after the process of harmonizing the data set, following the same example that I showed before with the non-natives, we have two curves, and all of them are showing a slight trend towards more similarity. What I want to highlight is that full change, complete turnover, will be 1. Okay, zoom. We need to go to 0.2, the maximum. So it is not that is a max, super. It's a change towards similarity, but it keeps being a lot of space to be this turnover and this similarity trend. Okay, and all this, what is bringing us is like, okay, we showed that there is a trend towards similarity in all the islands, but we haven't start trying to understand this trigger of people. So what we did is basically checking eventually in each of these time beams if the island was settled or not. Okay, and this is how basically this plot is showing. So when the time beam of both islands were not colonized, that would be the box plot. When one of the islands is colonized and the other not, that would be the similarity. But when both time beams, we have evidences the islands were settled, this pairwise record the similarity index increase. So it's suggesting that, yes, people have the key potentially to start understanding the similarity trends. So basically, we are ending the talk with these conclusions, and we say that basically the site comparison showed that floristic homogenization began between 3000, and it looks asynchronous with the timing of human colonization. We see as well the pairwise Ray-Curtis similarity results also show that when two islands were settled in a given time interval, the similarity is greater. And a weak conclusion, but it's something that we like to explore, is that high elevation sites tending to show less floristic homogenization. And these sites as well were the ones that they are not following the turnover, the acceleration of turnover, suggesting that these are particular sites and probably something really interesting to keep studying. And as well, we showed that to Jarova, again, the illustrator, and he created this illustration for us. And that would be his representation of the main results. And they say, guys, let's stick around and avoid mingling the islands, the highest bits of the islands. And where we are based. So we are based in La Autónoma CREAF. And this is the faces of the team. And we are the Temporal Ecology and Biogeography lab. And basically, these are the faces of people collaborating in this project. Maybe we can add some other faces today. So thank you. Thank you so much.